a very good audience for Charlie to present to. So it is really my great pleasure to stand here today to introduce Charlie. Before I do that, I'd like to make the acknowledgement of country. So I'd like to begin by acknowledging the traditional owners and custodians of the land upon which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present, and the elders from other communities who may be here today. So Charlie joined my lab in 2014. So his PhD has been going really for about 3 years. And at that time, the insectary was just beginning to produce infected mosquitoes, not really in a high throughput way. It was actually very variable. And Charlie had been quite interested in the project or projects that we're offering, but was really thrown in the deep end in terms of getting a facility that wasn't really up and running with help from others as well, of course. But Charlie's been really dogged in his thinking and his approaches to trying to get this facility to work as well as it can, and it's a real testament to his contribution as well as others in the lab and also in the facility. Of course, a large number of people who contribute to making it work very well. Charlie received his master's degree quite a long time ago at the London School of Hygiene and Tropical Medicine. He was conducting field research in Uganda on schistosomiasis. Yes, fantastic. I should have practiced this a little bit more, I think. He then worked on HIV and STI surveillance and prevention for 3 years at the Health Protection Agency in Cambridge in the UK. So you can see a theme here. He's from that island or colony up north that is now no longer part of Europe that voted this year. So in 2011, he was, I'm reading his bio that he sent me, he was convicted of shoplifting and deported from Britain to an outpost in Australia, joining the colony in Australia, where he took up a research assistant position here at WEHI, and he then investigated population genetics of malaria parasites in Papua New Guinea before joining my lab to work on the liver stage of Plasmodium falciparum. So without any further ado, it's a great pleasure for me to welcome Charlie up to give his PhD completion seminar today.
Hello. Is it working? Yes. Yeah? Okay, cool. Right. Well, thank you all for coming. So today I'm going to talk about the projects that I've been working on for the last 2.5 years since I joined Justin's lab. So basically I'll start with a brief background to malaria, then go on to talk about my first project, which is protein export and whether it's important in the role, plays an important role for gametocyte development and onward transmission to mosquitoes. And then I'm going to focus most of the talk on the role of thrombospondin-related sporozoite protein and the role it plays in human liver infection. So to start with, this is a map of malaria prevalence from 2010. And you can see that malaria is focused in the tropical regions of the world. And this is falciparum. And this map is shrinking, but it's still a massive problem in the developing world mainly. With approximately 400,000 people dying last year. These estimates vary, but that's a rough figure. And it largely corresponds also to where the poorest people in the world are living, primarily in sub-Saharan Africa here. And this graph here just shows a correlation between the percentage of a country's population that are living on less than $1.25 a day and the number of people that die of malaria. Per billion people. So you can see it's a disease of poverty, and it promotes poverty in these countries, and this is one of the reasons why I'm interested in tackling it. So it's largely refined to the tropics because the malaria vector species are primarily tropical, and the Plasmodium falciparum requires quite a high temperature within the mosquitoes to fully develop. But you can see that there would be transmission in Australia, indeed there used to be, and in countries like the southern US. But it's been eradicated from these countries because basically we're wealthy and can afford to do so. So I'm going to linger on the life cycle a little bit, apologies for that, but we do study the entire life cycle in the body lab. So there's aspects of this that you won't be perhaps as familiar with as just the blood stage, which is normally talked about here. So This starts with the infective bite of a female Anopheles, in our case, Stevens eye mosquito, which is what we have in the colony here. The sporozoites are deposited into the skin, not into the blood directly. They then traverse into the capillaries in the skin, make their way towards the liver, where they recognize Kupffer cells. They traverse through these into the liver. They traverse through multiple hepatocytes until they reside in one, where they form a liver schizont, which is up here in this part of the life cycle. Now, this is a massively replicative phase of the life cycle, and one sporozoite will go on to form approximately 40,000 daughter merozoites over the course of a week or so. So these eventually rupture into the bloodstream through merosomes, you can't really see here, but sort of packages containing merozoites. And then this starts off the blood stage of the infection, which is the patent phase, which causes all the symptoms of malaria. So they invade red blood cells, which is a sort of odd choice of cell, being devoid of any protein export or import machinery, basically just sacks of hemoglobin. And when they've invaded these cells, they then drastically remodel these cells through protein export, which is what I'll talk about in the first part of my talk. So they grow inside these cells, exporting proteins into the host cell, allowing protein, import of nutrients, export of waste, and also allowing this sort of sequestration phenotype where they become sticky and they stick to the microvasculature of organs, which causes some of the severe complications. So a small portion of this cycle takes 48 hours, but a small portion of these will go on to develop into the sexual stage of the life cycle, which takes 12 to 14 days and form these gametocytes, which are these funny sort of banana-shaped guys here. When they're taken up into a mosquito midgut, the drop in temperature and the shift in pH stimulates 3 rounds of mitosis in the male, which then exflagellates, so forming these microgametes here that are very sort of sperm-esque. And then these microgametes will then fertilize the female macrogametes, resulting in the fertilized diploid stage of the parasite's life cycle. So this is, in fact, the only stage which is diploid, is the zygote onward to eukinete. And therefore, mosquitoes are actually the primary host of malaria, not humans. We're just the secondary intermediate host. So this develops into a motile eukinete, which is this guy here. And this will traverse through, or is actively motile through the blood meal of the mosquito until it reaches the midgut epithelium, the layer of cells surrounding the midgut of the mosquito. It then traverses through this layer until it reaches the basal lamina, which is kind of like the bag, the membrane sac which surrounds the midgut. And there it will develop into an oocyst. And over the course of 10 or so days, 10 to 18 days in Falciparum, it develops thousands of sporozoites. So it's another sort of massively replicative phase here and here. And these oocysts eventually rupture, releasing sporozoites into the hemolymph, which is effectively the sort of blood circulatory system of a mosquito. And they make their way to the salivary glands of the mosquito, where they can then be transmitted on to humans. So we're quite interested in these steps here, the gametocyte transmission to mosquitoes, as well as transmission from mosquito to humans, as they represent massive bottlenecks in the parasite's life cycle. So it's very few gametocytes are taken up by mosquitoes, and very few sporozoites are deposited into the skin to initiate this phase. So if we could block either of these, this would be a massive big deal. And there you go, that's done. Right. So in order to remodel the host cell, the parasite exports hundreds of proteins into the host cell. So this is a blood stage model. So in the blue, you have the nucleus, green is the ER, and the purpley color is the parasite membrane. And then you have a parasitophorous vacuolar membrane, which again encircles the parasite. So it's within 2 membranes here, and this parasitophorous vacuolar space which surrounds the parasite. And it can export proteins into this space, forming PTEX channels within this parasitophorous vacuole, where proteins are unfolded and then pass through and refolded on the outside. And then they extensively remodel the host cell, laying down knob-like structures which anchor surface antigens. So this would be Plasmodium falciparum erythrocyte membrane protein 1, for example, which is a major antigen of Plasmodium falciparum. And these, so the cell is extensively remodeled, which allows for the aforementioned protein and sort of nutrient uptake, waste exchange, and also the sequestration of these parasites in the microvascula, which causes complications such as cerebral malaria, and is also associated with the very high parasitemias you get with Plasmodium falciparum. So one of the major and best understood pathways for protein export is pexel-dependent protein export, which involves the Plasmodium export element. So to explain this, I'll just use an example, knob-associated histidine-rich protein, which forms these knob-like structures to which the major surface antigens are attached. So this was work done at WEHI back in 2009, and this just shows the that protein I previously mentioned, the signal sequence in red, the pexel motif, which is a 5-amino acid sequence, R, X, L, X, E, Q, or D, and then a bit of downstream sequence linked to yellow or green fluorescent protein. And for the sake of simplicity, I'm just going to talk about these ones. So this is the wild type, and this is the triple mutant. So the first, third, and fifth residue are all mutated to alanines. And in this example, the wild type, you can see that the protein is exported just fine into the red blood cell. But for the triple mutant, there's no export into the host cell, and the protein backs up inside the endoplasmic reticulum. So the parasitophilus vacuolar membrane is labeled here in red. So this completely blocks protein export, this mutation of these key residues. So this is the pexel, and this is where it's cleaved after the third residue, which results in this new N-terminus of the protein, which is then N-acetylated and licenses the protein for export out into the host cell. So it was later discovered that there's an aspartyl protease responsible for this cleavage step, which is called plasmepsin 5, and this is what directs proteins out into the host cell. So more work done here at WEHI ended up with the production by Brad Sleeves and Tony Hodder of these peptidomimetic compounds, so WEHI 916 and 842. So these mimic the pexels, this pexel motif here, and bind in the active site of the enzyme, preventing the binding of normal substrates and blocking cleavage of these. So these little graphs at the bottom here show the activity of Plasmodium falciparum and Plasmodium vivax, which is another species that infects humans, plasmepsin 5 enzymes. And this shows the inhibition up here when they're in the presence of WEHI 842, which is the more active or better fitting compound, and WEHI 916. So this paved the way for using blocking of protein export as a potential drug target. And indeed, if you put these compounds into blood culture with Plasmodium falciparum, you see an effective concentration of about 400 nanomolar for 842, 5 micromoles for the slightly less effective compound 916. So this does kill parasites, at least in vitro. And this is the basis of ongoing work that's going on in Alan and Justin's labs. So with the use of 842, they were able to co-crystallize plasmepsin 5, and this is the structure of it here. And you can see the 842 molecule bound in the active pocket here. So it's important in blood stage, but is it important for gametocyte development? So that phase that I told you about earlier, which takes between 12 and 14 days. So to study this, we set up cultures which undergo 2 cycles, so that's 2 48-hourly cycles, from the ring stage through the trophozoite to the schizont, which ruptures, and then back again. And then at this point here, you get a really high parasitemia in the culture dish, and the environment becomes quite toxic. And then this sort of promotes the commitment to a gametocyte or a sexual stage, possibly because the parasite is trying to escape a toxic environment, and the only way to do so is to become a sexual stage which can be transmitted onto a mosquito. So we hit the very early gametocytes with drug for 7 days in the culture medium, and then for the last 6 days, we take them off this, and then, oh, wrong button. this is what the cultures look like at the end of this process. So these are healthy-looking gams, in my opinion. So this is about 2 to 4%, which is fairly standard for these cultures. And this is just the vehicle control, which is DMSO. With 1 micromolar concentration, you still do get stage 5 gametocytes, but you also see a fair few deformed gametocytes, and the percentage is far lower, commonly around here. And then with the highest concentration, you basically see nothing. So it's killed absolutely everything. So then I'm going to go back to a bit of intro kind of stuff. So to test whether those gametocytes that were produced in the 1 micromolar concentration are viable, we tried to transmit them to Anopheles stevensii mosquitoes. So these are the mosquitoes we have in the colony at WEHI. So that's a typical GAM culture. And it looks pretty gnarly, you know, it looks like maybe I sneezed in that and it's gone bad, but actually this is just what they look like after about 14 or so days. And these are the blood feeders. So this is a glass membrane feeder. We stretch parafilm over the top, and then that basically forms the skin on which the mosquitoes will then feed. Ta-da! And yeah. So this is a typical feed setup. So you have the water pump here, which is pumping hot water through this system, and all these little feeders here, and you put blood in them, and then the mosquitoes come underneath and feed. And as Justin mentioned, it was quite difficult to get this setup to work, and we had a lot of struggles to actually transmit falciparum to these mosquitoes. And in the end, it was they're an Indian mosquito from urban areas, so we just tried a few tunes, and they transmit a lot better in the presence of Punjabi MC. So this is just, you know, an example of a standard feed. And you can see they're loving the blood meal. And they perform a blood filtration step while they're actually feeding, and they leave this sort of nasty wee patch on the bottom of the cage because they're filtering out the nutrients as they actually feed. And our colony has this specific bum dance it does there, this guy, which is unique to WEHI. And there you can see some blood-fed females. And that's about it. And there's that diuresis step that I talked about. So it's filtering the blood as it's feeding. Okay, enough of that. Yeah, and so this is the breed room, which is kept at about 26. It used to be 27, but it's about 26, 27 degrees. That was a glove box, which we used to handle infected mosquitoes. And these are the double cages in which we house infected mosquitoes, just as an extra precautionary measure. And these are the colony cages, which just produce extra, well, we just breed from these. So they don't need to be double caged because they're never exposed to Plasmodium falciparum. So a little bit more on how we do this. So this is a female Anopheles here and here. This is a male. You can tell this bifurcated tip there. And then they also have this sort of bushy, moustachy antennae here, which is quite easy to distinguish. And I mean, it looks like I was drinking before I did this dissection, but I wasn't. I just had to use, just do it by eye because one of the eyepieces was taken up by the camera. So this is a bit of a spaz dissection, but you basically want to grab the second to end segments, and then you pull the midgut out here, and then you try and get rid of these Malpighian tubules here, this sort of stringy stuff, that's gone. And the midgut is there. Okay. I was going to try and do salivary gland dissections as well, but given how difficult this was, I just gave up. So that's the midgut there, and we dissect them out about 7 days after the blood feed, and then count the oocysts, which I showed you in that life cycle vid. There you go. Come on. So this is what they look like. So we take them out, we stain them for about 15 minutes in mercurochrome, and that's taken up by the oocysts and allows us to visualize them. And then we just look at them under a light microscope and count the number of oocysts. So that's these forms here. This is about day 7. They actually get a lot bigger than this when they're maturing. But you don't want to count them too late or they start to rupture and then it becomes difficult to actually count them. So with all that in mind, we then fed these to mosquitoes. So on the green are the controls, basically. So this is just with normal culture media. And then I fed them with DMSO as well, because not actually with the blood feed, but during this 7-day drug treatment, there was the DMSO control. Because I just wanted to see if there was any difference between sort of normally reared gametocytes and the ones in the presence of DMSO. And as you can see, there's no difference, so I didn't bother repeating this again. This compound here, 6025, is very similar in structure to 842, but it just doesn't bind in the active site of plasmepsin 5. So this is the true control. And as you can see, these gametocytes here are just non-viable. They do not transmit at all to the mosquitoes. And I did this again with a slightly lower concentration of 842, and you get a similar deal, like they don't transmit at all, but you're getting a slight recovery of transmission at the very lower concentrations. And this, if you remember back to that previous slide, is quite a similar sort of concentration of an EC50 of 400 nanomolar, which was killing the blood stages. So essentially, yes, protein export is essential for early gametocyte development. And I say early because if we do this experiment, I have data which I won't show here because I just don't have time, but we have data where we add the drug at later time points, and the later you add it, the more drug you need to have an effective killing of these gametocytes. So that's very early on, but what about right at the end? So I reared gametocytes in the standard culturing method and then just added 10 micromoles of 842 along with the blood feed. So I just spiked the blood meal that I put in those feeders with the plasmepsin 5 blocking compound. And you see no transmission blocking effect of this whatsoever, which is kind of makes sense because you'd think that in a 14-day maturation process, all the protein export would have occurred already and stuff would be packaged ready for export in these sort of mosquito stages already. So this wasn't really a surprise, but it was just nice to confirm. So in confirmation of this, so just summarize, it's already been shown that plasmepsin 5, well, pexel-mediated protein export is essential for sexual stages, but it's also, asexual stages, but it's also essential for the development of gametocytes and transmission to mosquitoes. And that blocking protein export early in development prevents transmission to mosquitoes. So this is quite nice, really, because there aren't many drugs currently used which have gametocidal activity. So this sort of makes blocking protein export or targeting plasmepsin 5 quite an attractive drug target to follow up on. So this is the second part that I'm going to talk about, the main meat of my PhD in Justin's lab, the role of thrombospondin-related sporozoite protein. So it's quite a small protein, about 17 kilodaltons in size. This was from a paper where they aligned it, and you can see the signal cleavage, signal sequence peptide cleavage here. And it has a putative pexel motif. So it's misaligned here, but it's conserved across all parasite species. So these are the human ones, Falciparum and Vivax, Knowlesi, Simonolgi, and monkey malarias, Berghii, Archibaldii, and Uelii are rodent species. So it's conserved across all of them. And if you take this pexel sequence, the RLLNE, and you take a synthetic peptide of that, incubate it with plasmepsin 5 in red, you see a size shift, indicating that the pexel is indeed cleaved. So it seems like it could be a real pexel motif. And then there's data from this paper, Kaiser et al. 2004, where they looked, this is an immunoblot using anti-sera raised against TRSP, and they find two species here. And these roughly correlate to the sizes that you would expect if this was indeed a cleaved pexel. So these are oocyst salivary glands and, oocyst sporozoites and salivary gland sporozoites. And this is especially interesting because it's been shown that hepatocytes that are infected with malaria are actually resistant to apoptosis. And it's sort of postulated that this might be because proteins are being exported into the host cell and then messing up the apoptosis machinery within the cells, sort of skewing them for a more pro-survival sort of state, which would support the development of that really massive, sort of unusually huge parasite growth within the liver. So much to Ethan's delight, if he's here, there are glycosylation sites present on TRSP. So there's a Seaman oscillation site and no fucosylation site. And there's a conserved R2 plus region within this thrombospondin repeat. So this area here, from about W on down to sort of C here, is known as the thrombospondin repeat. And these are commonly associated with adhesion and invasion. And indeed, they're common as well in other two really well-classified proteins of sporozoites, circumsporozoite protein, which is currently the target of the only malaria vaccine which is available, and thrombospondin-related anonymous protein, which is involved in the invasion of salivary glands. So these are several reasons why we thought it'd be interesting to check it out. So that's the basis of where we started at. So Matt O'Neill in the lab knocked it out of blood stage, and in line number 3 here, you can see the hybridization of the correct size in the 5' flank and the 3' flank, confirming that the parasite, the protein has been knocked out and the integration event was successful. So these were then cloned out to produce E7 and G5, and for the rest of the talk, I'll be referring to them as KO1 and 2. And this is, it makes sense that we can knock it out in blood stage because there's, from the previous data, again from Kaiser et al. 2004, they see no expression in blood stage, but it's highly expressed in salivary gland sporozoites here. And again, by QRT, RT-PCR, you see high expression in oocyst salivary gland, oocyst sporozoites and salivary gland sporozoites. And when we do a blood stage growth assay of this, we see no defect in growth. So this is just a one-cycle growth assay, and you see about a 7-8-fold replication in the wild type here, and you see roughly approximately the same. At least there's not a defect in blood stage growth. So then how does it transmit to mosquitoes? We can read gametocytes just fine, and they look healthy. They also transmit to mosquitoes just as well as wild type. So these oocyst these transmission experiments are notoriously variable. So you can see the x-axis here varies considerably, and that's just a feature of working with these really annoying mosquitoes. So it's inconsistent, but there's no real, this is just 2 lines of wild type fed on the same day, so you can see the difference between just the wild type alone. But when you compile all these data, you see there's no defect at all in the infection of mosquitoes. So there's no defect in transmission to mosquitoes, but what about growth within these oocysts? Do they produce viable sporozoites which can then infect? So these are the mean counts of pools of mosquitoes dissected. The salivary glands are taken out. We mash them up with a pestle and mortar and then count the sporozoites under a hemocytometer, and from that you can calculate how many sporozoites you have, especially when you go back to the number of mosquitoes you dissected. So here you see there's no defect in sporozoite production, and these made it into the salivary glands, so they can obviously invade salivary glands. I've normalized this for prevalence, so this is using the oocyst data. I mean, really, this just gives perhaps a slightly more accurate view, but again, there's no defect. And then if you look at the number of mean sporozoites per oocyst, so that's using the oocyst data from the week before when we were dissecting, and then the sporozoite numbers on day 17 or 18, you can use both of those, the oocyst as the denominator, to then determine how many sporozoites you get per oocyst, and it's approximately the same. So there's no difference here. So in terms of where the protein is localized, this is data from, again from Kaiser et al. 2004. This is the Bergii rodent model of malaria, and this is thrombospondin-related anonymous protein, circumsporozoite protein. So these are the other 2 main sporozoite proteins that have been really well studied. And you can see here in green you have TRSP. It shows this kind of apical orientation, which they claim to be bilobed, a rhoptry localization. I can't quite see that, but okay. And yeah, so it seems to be apically orientated, but Matt O'Neill in our lab GFP tagged the protein, and we see a quite different localization where it seems to be surface localized on the surface of the sporozoite, which looks a lot like the CSP localization. And then earlier this year, a paper, they labeled surface exposed proteins and consistently found that TRSP was in this fraction. So it's very likely a surface protein, which makes sense given it has a transmembrane domain. So with all that in mind, we were wondering, would they traverse through human hepatocytes? So far there seems to be nothing wrong with them. They infect mosquitoes fine, they develop in mosquitoes fine. Do they traverse through hepatocytes, which they would have to do to infect mosquitoes? So this is my cartoon of a traversal assay. So we have HCO4 cells, which are human hepatocytes, which are permissive for Plasmodium falciparum infection. We incubate them with FITC-labeled dextran and sporozoites for 2.5 hours, and the sporozoites will traverse through cells, rupturing the membrane, wounding. The cells reheal and then labeled with FITC-labeled dextran. We can then trypsinize these cell layers and FACS sort them. And this is what you get after 2.5 hours. On the left is just the control, which is with dextran, and on the right is the wild-type parasites having traversed through these cells. So you get around 20 to 30% traversal in your wild-type, and this is normally, well, it depends where you set the gates, but in the low sort of single digits. And I did a time course to see how long the sporozoites are actively traversing hepatocytes for. And you can see that traversal is over at about the 2.5 hour point here. So for all the traversal experiments we do them for about 2.5 hours. And this is typically what we get, or what I've been getting for these TRSP knockouts. So you have the dextran down here, about 25% traversal for the wild type, and then there's a consistent mild defect in traversal in the knockout parasites. And when you quantify that as a percent defect, so up the y-axis here, you have the percent that these sporozoites are defective as opposed to their wild-type controls that were run in the same experiment. You can see there's consistently around about a 30% defect in traversal of these parasites. So this is the first evidence that this protein is involved in something to do with traversal or invasion of the cells, which ties in with that thrombospondin repeat being involved in invasion and adhesion. So to check whether this traversal defect corresponds with an invasion defect, we do a similar assay where we have a monolayer of hepatocytes. We incubate that with sporozoites for, well, it depends how long you want the assay to run for, but between 3, 24, or later time points. It becomes difficult to analyze at later time points because the monolayer just keeps growing and growing, and then you end up with sort of vanishingly small returns. But at 3 hours, this is an example of a 3-hour time point when traversal stopped, so it should really be invasion events. We stain once with the anti-CSP antibody, which is that surface protein I talked about earlier, and then a secondary in red, and then you permeabilize all the cells, stain them with the same primary, and then the same green secondary, and you can see some of them are green only, indicating that they're inside and not outside. And then you can quantify this and then get your percent invasion. So at 3 hours, rather contrarily, we don't see any defect in invasion, which doesn't really tie with the traversal data that I have. But so this is experiment 1 in brackets 2 and 3, and it's 3 wells quantified in each experiment. And this is just one of the clones, and this is as a percentage of the hepatocytes invaded, so as a percent of hepatocytes counted. So it's between 1 and 1.5%, it's quite low. So we add the sporozoites in a ratio of 1:1 to these, so it's quite an inefficient process in vitro. You can then look again, but quantify this as a percent of the sporozoites that were put onto that were counted. So you can see it's a much higher percent. So about 25% of the sporozoites you count are actually inside the cells. And what's interesting here is that you actually do start to see a slight defect. I mean, it's not statistically significant, but there is a consistent defect here as a percentage of internalized sporozoites as opposed to when you use hepatocytes as the denominator. And then just to confirm that I did a fair job, so all these are counted blinded, and then I find out the result at the end. I just quantified the number of sporozoites that were counted, and that's consistent across replicates, and then the number of hepatocytes that were counted, and that's also consistent. So these data are quite sort of robust, but they are not in agreement with what was previously published for Plasmodium berghei. So they've done the same experiment basically with their mouse model, and they find between a 60 and a 40% defect. This is the defect up the y-axis, and this control here is just a compound which totally blocks motility, so they won't invade. They also find an increase in partial invasion. So that's, you see the red part of the parasite is outside the cell, it's co-stained in green, but then also there's a piece of it here. I mean, the IFA is a bit iffy, but it's actually inside and outside at the same time. So it's partially invaded. I looked for that in my experiments, and I just couldn't see any. So then I looked again at a later time point, so 24 hours. And there's no defect at all, so the invasion events that have occurred are legitimate. If anything, I mean, losing the gene is actually beneficial by the looks of it, at least here. So this is just one replicate. I have more to count, but the number of sporozoites counted is approximately the same. Hepatocytes counted is the same. So as an additional thing for this, to really confirm that these 24-hour invasion events are legitimate, I wanted to quantify the extent to which they are developing. So this is a slide from a paper showing the development of sporozoites, how they develop over time in an environment like a hepatocyte-like environment. So they start to sort of ball up, and then the sort of two arms of the sporozoite, the sort of tip and the rear of it, sort of get sucked into this round body, and then they become round. So I quantified what I determined as rounding as anything that looks like this, sort of onwards. And of the percent of sporozoites that are inside cells, there's no difference in the percentage of them that are actually rounding up. So there doesn't seem to be a defect in this sort of early developmental phase. So far it's a traversal defect that doesn't correspond to an invasion defect in vitro, but these are in vitro assays, quite artificial. So the next step, we stepped it up a notch and went to humanized mice. So these are skid mice, and they are transgenic with a urokinase-type plasminogen activator, which is driven off an albumin promoter, which makes that expression liver stage specific, not liver stage, liver specific. And then they're repopulated with, they're injected with primary human hepatocytes when they're pups. And these repopulate the livers, making them competent hosts for human diseases such as hepatitis and malaria. So what we do, we take these mice, they're sent to us from Norman in Canada. KMT Hepatech is his company. And we basically just inject them with our sporozoite lines and see what happens in the livers 6 days post-infection. So this is what one of the chimeric livers looks like. You have this sort of mottled appearance of this pale and dark tissue. And depending on who you talk to, they'll say, like, the dark is human or the dark is mouse or whatever. But you can see that it's chimeric. And we cull these mice 6 days post-infection, so if you recall that video, that's about as far as you can go out in this developmental stage before they would start bursting out into the bloodstream. We cull the mice at day 6, and we chop up their livers, saving half of it to make OCT frozen sections for microscopy later on, and then the other half we homogenize and extract the DNA from, and we run qPCRs on those to quantify liver load. So we run a qPCR on the percent human chimerism. So this uses a housekeeping gene from both a mouse and a human against one another. And you can see that we have fairly consistent between 40 and 20% chimerism in these mice. In the blue, you have the mice that were infected with wild type, and in red, the mice that were infected with the knockout. So it's roughly the same across groups. But you see a massive defect in liver stage invasion, liver stage burden 6 days post-infection with the knockout parasites as opposed to the wild type. So this is number of parasite genome copies per million hepatocytes. And you can see, for the most part, the wild type parasites produce good infections, whereas the knockouts are consistently extremely poor. And this is sort of in agreement with the previous WEHI data that I would classify as preliminary. But yeah, so it kind of ties in, but we're seeing what a much stronger phenotype than they have done in this WEHI model. So it begs the question, you know, is this defect here? Is this, defect here? Is this a result of a growth defect, or is it this inability to actually invade cells in the first place? Because at least with the in vitro data, there's no evidence that would suggest that this is an invasion protein. So maybe it's a growth thing. They're getting in, and then they're just remaining really small, and they're not multiplying properly. Because like I said, you get 40,000 copies of the genome per liver schizont in a successful liver stage parasite. So I took those OCT liver blocks, sectioned them, and did IFAs of them. And you can see this granular appearance of these liver schizonts here with the DAPI staining showing the masses of nuclear material dividing up into these thousands of daughter merozoites, which will eventually rupture out into the bloodstream. So I drew around them, effectively measuring the size, and from this you can get the area within each liver schizont. And again, this was done blinded, but it pretty soon became obvious which was which. So these are the sizes of the liver schizonts day 6 for the wild-type mice, in the wild-type mice, and this is in microns squared. So they're consistently, there's variation, because obviously you're not always going to slice exactly through the middle of a liver schizont. And then for the knockout, we see basically the exact same size, but there's just far fewer of them. And then to confirm that I hadn't biased this by looking at different areas of liver tissue, I measured the area of tissue I looked at. And you see I've looked at, about 7 or 8 centimeters squared of tissue here, even more than for the wild types, yet there's far fewer of these. So if you take the number here, the area here, you can then work out the number of liver schizonts you have per centimeter squared of tissue, which is in quite nice agreement with this qPCR data from before. And in fact, I'm going to have to repeat this PCR because I'm not sure about this guy. Another thing I did just for funsies was to check whether the development of the schizonts was the same across different lobes of the same mouse. And you can see it pretty much is, with the exception of this lobe here. I'm not a liver expert, and the livers are so weird looking. I don't know which lobe corresponds to which, but these are different lobes. So there's a massive defect of invading the liver. What causes this? So is it something to do with the chimeric nature of that tissue? So there's human and mouse tissue in these chimeric livers. They already have a slight traversal defect. So is it exacerbated by the presence of mouse tissue, which they then can't actually traverse through? Maybe TRSP is binding a specific human receptor, which isn't present on mice cells. Or is the motility is the deficiency in motility? So to test this, I went back to that traversal assay that I explained earlier. And this is just more replicates of the same thing. So you have H indicates mice cells that were being traversed here and here. And M indicates murine cells. So these were HEPA-1/6 cells. And you can see that there's a consistent defect in the traversal in this sort of aberrant species mix. So they don't like traversing through mouse cells as much as they do human cells. But this defect is fairly consistent across all the... so this is a % reduction of traversal from human to mouse cells. It's fairly consistent across the parasite lines. But still, there is this defect, and these assays are in vitro, right? So they're just on a sort of flat level. They're traversing through a handful of cells. When you're actually in a liver environment, every cell you traverse, you're just encountering a new cell, and then another cell, and then another cell. So it's possible, I think, that this sort of mild defect that we see consistently across all these traversal experiments is potentially responsible for this defect in vivo. So one of the last experiments I've been working on is motility, and really this should have been done a lot earlier, but I'll explain why I didn't do it earlier, because this experiment, the methods available are just a bit crap. So this is the traditional way of analyzing this. So you spin sporozoites down onto a slide, you allow them to move around for half an hour at 37 degrees, and then you fix them, label them with CSP, which lights up the trails they leave behind, and then you try and quantify this somehow. But given that the protein I'm looking at is localized on the surface of sporozoites, and it's commonly involved in adhesion, it's likely that you'd totally screw up your denominator when you were actually processing these samples, because lots of these sporozoites are going to wash off if there's a difference in adhesion between your wild type and your knockout. So at least in this experiment, they quantify it by the number of circles. So 1, 2 to 10, greater than 10, and then as a percentage of those that are gliding. But you can see it's a bit of a mess, and it's really quite difficult to quantify. Also, this is an extremely artificial environment, so sporozoites aren't commonly just sort of going round and round in circles in the liver. They only do it when they're in contact with a flat surface, and they always go the same way. So here's parasites that are going counterclockwise, and here's parasites that are going clockwise, and they just go round and round in circles. But when you put them in a more natural environment, say skin, there's no preference for the direction in which they move. And this is a result of this microtubule structure, which is slightly chiral, which only allows them to move in a counterclockwise way when they're on a flat surface. So I'd like you to concentrate on the quality of the movement of this sporozoite here. So this is Plasmodium berghii in a mouse liver, so intravital imaging. And you can see that it's quite an active, sort of all-over-the-shop process. And it's very much not going round and round in circles, except right now. So this is what I wanted to recreate in a motility assay in vitro. So to do this, I bugged people on lab help, got my hands on some Matrigel, which sets to a sort of gel-like consistency at about 37 degrees. And then I mixed that with the sporozoite mixture and bovine serum albumin, which activates the sporozoites to think they're in a mammalian host. And the result is I put this mixture on a slide, I put a cover slip on it, so you have a thin sort of 3D layer of gel, which they can move around in. And these are just this sort of chunk here. I couldn't play the whole thing because it's just too big. But this is on the Axio observer, which has a massive sensor, so I can capture loads and loads of events at the same time, which is absolutely really handy. So here is the central slice of a Z stack. And here is the minimum projection, minimum intensity projection of that Z stack. And you can see from the way they're moving, it's very much like that motility that you saw on the previous slide in the mouse liver. So I'm pretty happy with the way this has worked out, and it's sort of recapitulated the in vivo environment quite nicely. But then the next challenge is how you quantify this data, which is what I'll get to next. So I've been working with the computer wizard Lockie on, well, he's been working on this really, on automating the image analysis, the 2 terabytes of image analysis, image data that I have. So here you see, this is just sort of preliminary sort of trials of how we were doing things. So these are the automated trail tracks, and that's in the absence of BSA. So there should be a roughly similar number of sporozoites on each of these. I obviously can't count them, but it's the same mix that's going on each slide. And then when you introduce more BSA, you introduce more motility in the sporozoites. So that was the sort of step 1, so getting somewhere with the motility. And then he produced these, what I call tornado plots. So they show on the X and Y axis, you have the motility. So we had to flatten all the stacks because there was no way of getting the Z information out of them. So it's 2D motility, but in a 3D assay. So it's a lot better than the old one. And on the Z axis here is time. So these videos were about 7.5 minutes. And this is one with a fairly high concentration of BSA, and this one has a low concentration. And just as a sort of proof of principle, you can see that the tracks here often end up in vertical lines, and then they move around a bit, and then they end up stopping again, whereas these are more consistently motile. And the chimney here is a bit looser than this sort of tight central area here. There's also a lot more motility going on here as opposed to here. And this is because lots of the sporozoites on the slides without BSA just don't do anything. And you can flatten those and get sort of point of origin analysis of these. And that's unfortunately as far as I've got with this motility part of the project. So I have all the data there. We just need to figure out exactly how to analyze it. So that's a work in progress. So the final part of the project, which I've been working on in the last sort of month or so, is knocking the gene back in. So complementing the gene back in is the best way of showing that it's actually that which is responsible for the phenotype that we see. So in green is the knockout sequence. So the 5' flank here, TRSP would usually start here, but it's been replaced with a promoter, a drug resistance cassette, the terminator, and then the 3' flank. And then we used CRISPR-Cas9 to do this, and we found a guide that cut just upstream of where TRSP would start. And then this repair plasmid here has the 5' flank, which matches here. TRSP goes in full, And we either have a mutant or a wild-type pexel here, because, I mean, I talked about pexel export at the beginning, but we haven't really properly investigated it yet with TRSP. So that's what's coming up in the next part of this project. It's then either tagged or not tagged. Then there's a terminator, another promoter which drives another drug resistance, a terminator, and then the 3' flank, which actually matches this 5' promoter of this drug cassette. So you get this sort of action happens here. Yeah, so we're knocking, well, I've actually done these and they came up in 9 days. It's pretty awesome. So we had knock-in, so 4 lines have been made. So there's the knock-in, and it's at the endogenous locus as well. So it's being driven off the normal promoter, not off some artificial one that we're using. And then it either has the wild type or the pexel mutant, and then either of those either have an HA tag or they don't. So there's 4 constructs, 4 parasite lines. And these allow us to answer some of the questions which, are unanswered. So we'll be infecting, we've got these humanized mice in at the moment, and I'm rearing gametocytes of all these different lines at the moment as well. So we're going to be infecting with wild type or knockout. And the idea is that the knock-in of the wild type should infect at a similar level to the wild type. And then we don't really know what the mutant is going to do. I mean, given the surface localization and the transmembrane domain, it's unlikely, in my opinion, that this really is a pexel protein, but weirder things have happened, so it'd be good to see. And then the HA lines will allow me to do Western blots of these sporozoites to see if there's a size shift in the protein indicating pexel cleavage or not. And I'll also be able to do in vitro invasion assays in hepatocytes and look for export or otherwise. And I'll also be able to do IFAs of the liver tissue of these mice. Although at day 6, as you saw, the cell is totally full of parasites, so it won't really be possible to see if anything's actually exported or not. And obviously I have to quantify the motility data. So that just about wraps it all up. So in summary, I've shown that TRSP is important for the establishment of robust falciparum liver stage infection. The defect is not in growth, but in invasion. And this defect is only visible using sort of the in vivo model of these humanized mice, without which you don't really see any invasion defect or growth defect. So I think it's possible this mild traversal defect is responsible for this severe invasion defect in vivo, but we can't confirm that. And then the 4D analysis, 4D assay for Plasmodium falciparum motility is sort of optimized, and we just have to finish analyzing that. I just talked about that. So I'd like to acknowledge a lot of people. So Justin for taking me into his lab and sometimes dressing like me. No, so it's been a really great project. It's awesome working in the insectary, and it's just, I'm very grateful to Justin for taking me into his lab. Thanks to Alan, Jeff, and Chris for helping to guide the project. They're members of my PhD committee. Matt, who generated basically all the transgenic lines that I've been working with up till now, and just helps a lot with all sorts of angles of the project, old mate. Sash, who is heavily involved in the Gamete project, and again is a massive help around the lab, and I learn a lot from these guys. Jen was a postdoc in our lab and basically showed us how to do the vast majority of the mosquito stage stuff that we do. Annie and P-Man. Sarah and Mel are heroes in the insectary, pumping out 2 batches of Anopheles stevens eye mosquitoes every week of the year. And Sarah's grown accustomed to the fact that if you put an Englishman in a 27-degree room at 80% humidity, you sometimes end up with this dude. But she handles it admirably. So Brad and Michelle for producing the plasmepsin 5 blocking compounds that we use for lots of the experiments. Obviously the imaging guys, especially Lockie for a lot of the automation, Mark for helping set up that motility assay, and Kelly for help with various other projects that I've been working on. These guys, Tony, Ethan, Alan, and James, who've helped express TRSP protein, so I've got that in the pipeline as well, so we're making an antibody against it. Mark, Greg, and Liana that are involved in an apoptosis project that I've not had time to talk about today. Norman for the mice. These are other projects that I didn't have time to discuss. So Maria in Portugal's helped with an Ukani project of ours. Roll and Rebecca at Johns Hopkins have helped optimize that invasion assay. And then my previous lab and supervisors for all the help that they and the input for my previous sort of projects. Lab helpers for all the reagents that I bum off you. And then all WEHI, obviously, for being an awesome place to do this research. There aren't really many places in the world that you can do this kind of Plasmodium falciparum liver stage stuff. So it's pretty awesome. Melbourne Uni for my scholarships, various institutes I've visited. ASP for conference funding. Aussie Malar for a research exchange visit to see these guys. And then Justin's funding bodies. And thank you for coming.
Charlie's really accurately shown you how hard he thinks, how hard he works in chasing the truth. So really a fantastic seminar, a real indication of how hard he works in the lab. So he's timed it perfectly so that nobody can ask any questions. Now, if we have some questions, please give him a good grilling. Jeff.
Charlie, you alluded to the possibly confounding effect of having surviving mouse hepatocytes that your knockouts are trying to traverse.
Yeah.
I haven't quite thought the experiment through, but can you knock in mouse TRSP into human knockouts and see if you've got plenty of time left?
Oh, yeah. Only takes 9 days, yeah.
Would that work?
Yeah, maybe it would. We could at least do it in the in vitro assay. That would be interesting. Because the defect there is obvious, the traversal defect is consistent across all the lines. So we could whack in the mouse one and see if it just traverses as much in the mouse cells as it does in the human cells. Yeah. But yeah, I don't have time. But yeah, good idea.
So the knockout parasites, you've got quite a big difference between the in vivo data and the in vitro data.
Yeah.
Do you think part of it could be the flow, the blood flow, is getting, like maybe the response to the liver as opposed to being able to attach on and being.
Yeah, it's possible, because I haven't looked at how they traverse through macrophages. So Annie is also in the lab. She has a more sort of obvious traversal defective parasite. And they looked at macrophage traversal. So we could do a similar thing to see if because it's the Kupffer cells, or at least there should be Kupffer cells in these mice, you think, that it's adhering to first to then enter into the liver. Yeah, it's a possibility. I don't quite know how we'd answer it. Yeah, without maybe just that in vitro macrophage assay to see if they can't traverse those, or if it's more severe. I'm quite happy with the model of it just being a build-up of like traversal upon traversal, and it's just that sort of minor defect that becomes a massive thing when they're in a sort of dense liver environment.
You talked about your localization data conflicting with that, at least that interpretation of it being a rockery project. Is it possible that conflict's just a stage-specific thing, that less mature sporozoites, it's in the rockeries and then it gets secreted out onto the surface?
Well, I mean, there's salivary gland sporozoites just like mine. I mean, I can only I'd have to go back and read, but I mean, they would have just dissected them out of the salivary glands and done the IFAs just like I did. So I don't think, unless they're putting something funky in their medium, which is promoting changing the localization of the protein, I don't think so. And also there's that, it's got a TM, it stains like it's all over the cell. Yeah, I don't think it's...
Proteomics data is really quite strongly convincing as well. Like you shave the surface off and pull off proteins from the surface. It's abundantly there.
Yeah, I don't think it's in the rockeries. Yeah.
Alex?
Have you checked whether the parasitophorus vacuole is still intact in the knockout when it goes into the hepatocytes?
No, it's a good experiment. A difficult one, but no, I haven't. We could do that. So what, in the invasion assay?
In your invasion assay, because I think some of the other sporozoite genes that have been kind of essential, they get in, but they're so screwed up by the time they get in that they don't develop.
Yeah, so it could be sort of what we call aborted traversal, where they're just sort of traversing and then they give up when they're actually inside a cell. The rounding data that I showed kind of don't really agree with that sort of traversal abortion sort of hypothesis. But yeah, we could look. But it's a tricky experiment because the later time points, you have the hepatocytes just growing like crazy. The best thing to do, well, no, in vitro, yeah, we could do it, but we just stain with a PVM marker. Yeah, I haven't done it though.
So we might take one more, and then we're having some drinks and nibbles outside if you want to ask Charlie any further
Yeah, sorry for
questions.
everyone.
So one more to do.
So with A42 and gametocytes, you said that the older the gametocytes, the more A42 you need to kill the
Yeah.
gamete. What's the basis behind that? Is the parasite producing more plasminogen 5, or is the drug just not getting into the cell?
I think it's, I mean, this is my hypothesis. I don't actually have data on this, but it's just, if you look at the sort of, the very early gameto rings they form, so they look a lot like ring stage parasites, and that's when a lot of export's happening. So if you hit a parasite at that stage, it's not going to be able to export the proteins required. When it's already done a lot of exporting, it's going to take a lot more to kill the parasite. So it's kind of like a slow down. There's a lot of export occurring early on, and then it sort of wanes. And then to kill the parasite, you have to block sort of all of the protein export later on. But earlier on, if you block half of it, you're still blocking a lot of actual export. Sorry, I've not answered that particularly well, but there's more export occurring earlier on in gametocytes, and that's why blocking it earlier on kills them, and later on doesn't.
Okay, so we'll wrap it up here. Join me in thanking Charlie.